Well, good afternoon, everyone. By my clock, it is 1 o'clock here, so we'll go ahead and get started so we stay on time with our webinar today. I am Norm Groot. I'm the Executive Director here at Monterey County Farm Bureau, and welcome you all to our first of 3 webinar series on hosted by Ag Neighbors, California Ag Neighbors, or CAN as we call it in abbreviation. Thank you all for joining us today, and I also want to thank California Farm Bureau Federation for their gracious hosting of this particular webinar series and allowing us to use their Zoom platform. Also thank Don Steckel at CDFA and Tim Carden at California Farm Bureau for putting all the logistics together for these webinars. Really appreciate all the efforts in making this happen for us. At this point, I'm going to turn it over to Jim Houston, who is the California Farm Bureau Administrator, for a few welcoming remarks.
Thank you, Norm. Appreciate all your work on this, and thank you to all the panelists and participants today. The California Ag Neighbor Dialogue is really, really important. Both our experience here at Farm Bureau and a CDFA can tell you, the different commodities and different types of operations here in agriculture, the diversity that we enjoy also presents this opportunity and this need to have dialogue so that we can settle things through, time-honored dialogue, using our words and understanding where people are coming from and trying to solve those problems. It's something that ag is built on, for generations upon generations. This is how we've worked out our issues. And, in the 20 years I've been here in California, I've been fortunate enough to see, what seemed like intractable problems. Folks got together and sat down in a room, or a virtual room, I guess, in this case, and hashed it out. So it's very heartening to see all y'all. Appreciate the work that you're about to put in, that you have already, and look forward to a good dialogue. We've got a great group of panelists today, and we hope you enjoy their presentations. Please ask questions, share your thoughts, and join us for the next series of workshops. It's your input and your dialogue that makes this thing happen. So once again, thank you to all the people that made this happen. Thank you to the panelists and the participants. Thank you.
All right, thank you, Jim. And at this time, I want to acknowledge my co-leader in the California Ag Neighbors process here, Natalie Kraut-Greenberg from CDFA. If you want to wave at us and say hello, Natalie, thank you very much for all your leadership on this. A few logistics here for today. We're going to encourage you to use the Q&A function rather than the chat function. I'm going to be monitoring for questions as the presenters are making their presentations, and then at the end of each presentation, out of these 3 presentations, we'll have about 8 minutes or so that we can address some of your questions. So if everyone could use Q&A rather than chat, that would be great. Everyone's going to remain muted during the course of this webinar, so you don't need to use raise hand. And if we don't get to your questions that are listed in the Q&A, we will get back to the CAN dialogue group with those questions and make sure that they're addressed. Today's webinar is being recorded, so it will be available for review at a later date also. Just to give you an overview of what we're doing at this point is CAN is presenting this series of webinars to gain traction on knowledge of specific areas of technical, scientific, and operational parameters that may expose leafy greens to contamination risks. It is the intent of this knowledge base that will help further refine the elements presented in our interim report, which was issued last week, providing insight into management practices that will be key to food safety on both sides of the fence. Today's 3 speakers will share the research and current knowledge of E. coli O157H7 in cattle and wildlife populations. The speakers will cover the challenges and opportunities to reducing shedding of E. coli in cattle populations, and will discuss the transference of pathogens among populations in the localized environment. Our first speaker is Dr. Michelle J. Russell of UC Davis. She's a researcher and also a program manager for the Western Center for Food Safety, an FDA academic center of excellence located at UC Davis. Her research interests are in food safety and veterinary public health, and she is part of a large multi-year longitudinal study on the ecology and epidemiology of E. coli 0-157H7, say that fast 3 times, please, and other S-Tec in the Central California Coast region. So please welcome Dr. Michelle J. Russell.
Thanks, Norm, for that nice introduction. And yeah, my bio is a little bit wordy. So hopefully my slides aren't too much. I'm trying to keep it at the right level for the audience. We're going to do questions at the end. So I'm just going to run through my slides right now. And they should tie in pretty well with Dr. Atwill and Dr. Meyer following my presentation. And we might have a little overlap, but I think overall we're going to be very complementary. Rob Atwill and I go way back in terms of doing research in this area. And from the time when I used to be at the California Department of Public Health and then moved over to UC Davis in 2008 to be part of the Western Center. So my topic today, and thanks again, Natalie and Norm, for the invitation. I'm very enthusiastic, needless to say, about the California Ag Neighbor group. I'm honored to be part of it, and I just really think what you're doing is where we need to be right now in terms of our concerns about adjacent land use and E. coli, O157, other pathogens, in our coexistence as ag neighbors, as well as our coexistence with our wildlife and conservation communities. So I'm going to focus on wildlife, but it will have crossover with domesticated animals, just the nature of the beast here. The background is that animals are potential reservoirs of big words, enteric zoonotic human pathogens. Basically, these are bad bugs that come from fecal material, and they can come from animal feces. In particular, E. coli 0157 is more associated with ruminants and animals than it is with humans. It's somewhat of a spillover and can cause infection while the animals don't get sick. And the little diagram on the side there shows what a zoonosis is. It's basically a disease that can pass from animals over to people, but it can actually go the other direction too. But we'll be focusing on those that we worry about that might spill over to humans and cause disease. And that can happen in terms of, I'm more focused on bacteria for this, but primarily, and parasites can be important too, but the main routes where you would get sick from an animal that has a bad bug in its gut would be ingesting undercooked game meat that got contaminated. So same as ground beef and other meats, game meat needs to be cooked thoroughly. Or you can pick up these pathogens. There can also be direct contact, such as there was an outbreak from E. coli 0157 linked back to elk in Colorado at a picnic area where there was a lot of fecal material where people were eating, and there was transmission. But today our topic will be the middle bullet, fecal contamination of the food supply or water sources. Most of these pathogens just live naturally in the gut. These are not like some of the other pathogens that cause disease, like tuberculosis, brucellosis. Most of these animals, you can't look at them. And even in cattle in feedlots, they appear healthy, and they are healthy. These are essentially commensals for the most part. And again, these are fecal-borne pathogens, but we have on occasion found them in the oral cavity or on the fur, feathers, hooves, and the skin or hide of cattle in particular as a route of transmission in the slaughter environment. I'm just going to put a little self-serving pitch. Michael Doyle and I did co-edit a book that addresses this topic and has a number of authors, including Peter Dancek, who's been on the news quite a bit. He's at the center of the coronavirus research with bats. And so he actually wrote a chapter on the food linkage between bushmeat, wild animals, and foodborne pathogens. But now I'm going to switch to primarily focusing on produce food safety, and in particular our ag neighbor issues that are going on the coast. There are a number of different pathogens, but I will be talking about the Shiga toxin producing E. coli, particularly coli O157. And I'm glad I threw this in here because Norm very well put it. It gets very confusing what the taxonomy is. Is this a O157 or a 0157? And I'm not going to dwell on this for time, but the thing is that you're going to hear these names and these what we call serotypes. So it can be an O157 or these top 10 pathogens like O26 and new things being discovered. They all fall under these pathotype groups that are either called EHEX or S-Tec. So you'll hear those names thrown around either in reports or in regulatory context or in private labs sending results, but all of this may change. So it's great to learn it, but with whole genome sequencing, there could be some revolutionary changes of how we actually categorize these pathogens. I also want to say a word of how serious these pathogens are in terms of the linkage to wildlife. I kind of think of it as a low probability, high consequence event. But these can be deadly or cause permanent damage, the E. coli O157 and related strains. So it's very serious and something obviously we don't want being transmitted in the food supply. In terms of where is E. coli O157 coming from, I have at times make a hobby of doing a search to see if anyone has isolated E. coli O157 from a new animal. But it goes across the entire kingdom, all the way from humans to flies and protozoa. There are certain animals that more frequently harbor. So using the title of my talk that was given to me, that may actually be colonized and more frequently shed these pathogens. Others may pass through. There's a lot of things we don't know. These kind of studies are new and not easy to conduct. In terms, I do want to point out, wildlife are not our only concern in the produce production environment. Obviously, there are other transmission routes, and any of these can interact from watersheds to aerosols to the farm worker equipment and so on. But today, our focus for this webinar will be on livestock and wildlife. And then a few unique risks to produce to keep in mind for those who are not as familiar with the topic. We have a trend of eating raw, minimally processed and fresh cut bagged salads that have no kill step. So it's no longer the can of spinach from Popeye. It's we love our fresh spinach and other fresh vegetables, and that is very important to a healthy American diet. But these zoonotic pathogens can have a very low infectious dose, meaning you don't need to swallow much to get sick, especially a young child or somebody elderly or immunocompromised. And there is, while we don't believe that E. coli 157 goes up through roots, it's possible if it gets onto the outside of the plant or into the leaves, the edible portion, it might be able to cling there, to attach and not be washed off. And again, with our supply system, there's some potential for cross-contamination. The mechanisms are, as I mentioned, it can be direct transmission or in the environment. If there's fecal loading in the environment, it can then move to water, surface, groundwater, soil, sediment, bioaerosols. And that's a major area of our research at UC Davis, is how these pathogens move around from the animal gut into the environment and how they persist there. So our concerns, in terms of produce food safety and wildlife are the direct route with animal intrusion actually into the fields, and this has been observed on the pictures. That's deer feces on romaine lettuce, which will obviously be cordoned off and removed per the metrics and food safety metrics that are now in place for the leafy green industry. And below, Canada geese, which can be a problem because they travel in large groups and they make fecal material the size of a medium poodle, so they can make quite a mess. And they do get into strawberry fields, lettuce fields, and that has to be addressed with a corrective action. Flocking birds, again, they can be quite a concern in these environments, not just produce, but dairy and other ag environments and wheat crops that they can do damage to. They get into these huge numbers and can either spread disease or cause damage to the plants. And oftentimes in a setting like an orchard, they'd be causing damage to the fruit or nuts before you'd actually be worrying about a foodborne pathogen. But in some sensitive crops like leafy greens, you're going to worry with these animals being in the field, especially in larger numbers. And that's why we're here today. So I'm just I have so much data that some of it's published, some of it's not. Dr. Atwill will probably be sharing some of the studies that we did earlier on epidemiology and longitudinal studies over the years that looked at both wildlife and at cattle herds. And so I'm just going to highlight a couple of outbreaks that I'm familiar with and how they tied into wildlife, and then finish with a couple of recent studies that we did primarily with Center for Produce Safety. So, it's been known for some time that the deer and elk are reservoirs of E. coli O157. That was determined not long after cattle were identified as undercooked beef was identified, undercooked venison, and hunting like elk jerky has caused disease. And out in the environment, their issue is, they can also get into large herds and they can intrude into crop fields. Again, there are very few outbreaks that get all the way to root cause. Either the resources aren't there, the trace back isn't there, but there are a few examples where the root cause analysis went pretty deep. And Oregon Department of Public Health had an E. coli O157 outbreak in 2011 linked to strawberries. These were fresh strawberries from a smaller grower that had farm stands and a U-pick operation. And when they went out, they did find a lot of deer droppings in the fields. And they did find the same strain, the outbreak strain, in the deer fecal material mixed with soil in the strawberry fields that matched to the clinical strain. And there were 2 deaths and 4 individuals that developed kidney damage from that outbreak. So it was small, but very serious. The other large fauna would be feral pigs, javelina, which we've done a lot of work on in our research, and they can be a problem because the feral pigs are non-native. Most of the javelina are down south, and so you're not going to see them widespread, but feral pigs have become very widespread, and they reproduce fast, and they're a huge nuisance to native wildlife, native plants. And now there's documentation of them carrying animal pathogens as well as human pathogens, such as E. coli O157. This was discovered during the E. coli outbreak and environmental assessment back in 2006, when there was, again, a very intensive environmental investigation following this outbreak. And at the time, I was working for the California Department of Public Health on CalFIRT and was involved in the sampling out at the actual ranches. And the one that was ultimately implicated was actually in San Benito County. I know a lot of focus lately has been in South Monterey County, but this was actually more up north. And there were interesting adjacent land features that continue a theme since all the way to today, or hopefully not this year, but up to last year. Hopefully we're not going to have a problem this year. But, some observations is you have the valley area with the row crops, and then adjacent land being a cow-calf operation. In this case, it was a grass-fed beef operation, not a feedlot. There were a number of vineyards. There was a campground not far away, the San Benito River riparian habitat, and a lot of rangeland, all surrounding this growing area that was implicated. In terms of root cause analysis, the investigation was a couple months later, as it usually happens. The outbreak strain that matched the human clinical strain and the strain found in bagged spinach was found in a lot of different samples, suggesting widespread dissemination at this location. It was found in adjacent cattle feces, and fairly high prevalence of 33.8%. It was found in feral pig feces, as well as feral pig colons from pigs that were removed and euthanized on the property. It was also found in surface water and soil sediment, that was not used for irrigation, but had some potential for cross-contamination into groundwater. So it was pretty widespread. And it was apparent from the molecular typing. Now it would be whole genome sequencing. Back then it was PFGE and MLVA, but basically we had, there was a sharing of strains. Obviously it was moving around between the feral pigs, the water, the surface water sources, and the cattle, and who was moving it where. We don't know for sure, but there were plenty of opportunities for the contamination. And so this set off a lot of effort in terms of prevention and research of the livestock wildlife interface and produce food safety. One of the studies that we did that I'll highlight was with Center for Produce Safety, as well as leveraged with our FDA Western Center for Food Safety project funds, looking at the transfer of zoonotic foodborne pathogens from cattle on feedlots and wild animals to vegetable crops in the Southwest desert. Specifically, we were in the Imperial Valley and the Yuma, Arizona area, working with University of Arizona, as well as the Arizona Department of Game and Fish. And this was an intensive, almost 2-year field study.
Well, I think we have a frozen screen here. Apologize for that. We're going to wait a second and see whether or not it comes back.
Okay. So, so as I was saying, this study was fairly labor intensive, as can be the case with these field studies with wildlife. So the bottom line is that we did find a low level of E. coli O157, as well as Salmonella and non-O157, primarily among what we call the nuisance type birds, European starlings, cowbirds, red-winged blackbirds that do tend to aggregate on feedlots. And the feedlot prevalence was much higher for the non-O157 S-Tecs, and we also had E. coli O157, and are currently working on whole genome sequencing of these isolates to better characterize them. But the take-home message was that we did find that the rodents didn't travel very far off of the feedlot. There were barriers, and they just have a small home range, and they weren't that prevalent. They were, or they weren't that common. They were actually a higher density near the produce fields because the CAFO was not that friendly of an environment for a rodent. In contrast, the birds traveled regularly across produce fields to the CAFOs over buffer distances back then of 400 feet, but there's more complex reasons that, whether they would actually enter the produce fields and be of increased risk. So that data is still being analyzed. And another recent bird study, because there has been a lot of interest in birds, and a lot of the other mammals have already have been studied, and I think Rob will be talking about some of that shortly. But we did a year-long cross-sectional study at on the Central Coast, as well as in the valley at ag lands, and tested a lot of birds by capture recapture, and found that more evidence that these solitary type birds like this green heron on the left side really pose minimal food safety risks to human crops, are not where the attention needs to be. But again, we find these virulent strains of S-Tec and Salmonella from flocking birds, especially blackbirds, sparrows, geese. And a new finding from that study is we did isolate STEC from feet and feathers of a few wild birds. It wasn't common, but that was actually suggested by an industry person who had tested some birds with their private lab and had some suspicion that could happen. So that's going to be pretty insignificant unless you've got hundreds to thousands of birds, and then that can start to be significant. And we did find rangeland beef cattle were sharing subtypes of O26 and O163 with wild geese in this study. Kind of interesting in our PFGE analysis, and again, these isolates are going off for more detailed whole genome sequencing, but we had a few convenience samples that were found, not systematic testing, but beef cattle and river otter shared S-Tec subtypes, as well as geese. And we had the O26 from the feet, feather, and feces. We had O157 from a ruby crown kinglet, which was interesting. That wouldn't be a nuisance bird per se, but the spinach outbreak strain has actually been found by Fish and Wildlife in a survey years ago in dark-eyed juncos. And so it seems to be circulating around. This one from the ruby crown kinglet was not similar to the spinach, but we had feral swine feces that were collected on the Central Coast Ranch, and that strain was genetically very closely related to the 2006 spinach outbreak strain, which kind of ties into this whole mystery of the theme today of how are these things moving around, how are they persisting, and how are we going to control? Are we doing enough? Our studies generally support that the existing practices with trained harvest crews should be working, but there's still some transmission happening. So this is obviously an important part of FSMA's produce safety rule. There's actually an entire subsection on domesticated and wild animals, and we are embarking on the California Longitudinal Study, which Natalie is heavily involved in, along with FDA, CIFSAN, and our Western Center, to continue this, building on this work, addressing knowledge gaps in California, as well as a parallel study in Arizona. And I'm right at 1:30 and ready to send this over to Dr. Atwell.
You probably get up.
We're going to take a few minutes here to answer some of the questions that came into Q&A. So hold off just a second here, Rob.
Sure. You may want to stop sharing.
Yeah, thank you. Let's see here. Let's see if I can get back up to my first question. Well, let's just ask this question. What specific animals besides cattle might be involved in the most recent 2020 S-Tec romaine outbreaks on the California Central Coast?
So yeah, my knowledge is only based on the public reports that were released, and for the most part in those recent investigations, there were minimal testing of wildlife. It wasn't there were some observations, but in terms of systematic sampling, it was mostly described as scat. And so sometimes with outbreak investigations you don't have a lot of detail, and that's why we're doing the longitudinal study and have other avenues of research to try to get more information. I mean, the landscape down there has not changed much. There's still a lot of feral pigs. There's still bird pressure, rodent populations. But yeah, getting down to that root cause can be extremely difficult after the fact.
Okay, and moving on to the next question. We have recently seen at least one article, maybe more, specifically citing agricultural water as the source of contamination in the spinach outbreak. Can you comment on that narrow conclusion?
So yeah, that's it was a complicated outbreak politically, I'll just say it and be candid, but there were a lot of theories. It was unprecedented, the amount of sampling, and it was new. And I actually think we got a lot of information, even if not a smoking cow per se. Basically the outbreak strain was widespread. It was not found in well water or groundwater. But the CDC engineer hydrologist that came out was concerned about it was an older ag well with the potential for cross-contamination. So the contaminated pig feces and cattle feces could have gone through into surface water and contaminated groundwater. So we left that possibility open for sure. There could have been also direct contamination because of animal intrusions. And then the cattle were a mile away where we had positives, but there were cattle trucks going in and out right along the road, along that spinach field. So there were a lot of different potential routes. I stick to the fact that we know that the location, we know some risk factors, but we don't know without a looking glass back in time, we don't know exactly what that route was, but there were plenty of things to address. We learned a lot, I think, from that outbreak.
Okay, thank you. And then we have time for one more question here, and that is, can we discuss some mitigation strategies for birds that could be done on both sides of the fence?
So, right, birds are difficult, with pigs and deer and elk. It's fairly obvious. With pigs, you can do depredation, which may or may not be effective, but at least where they've been, and there are some options with fencing. With birds, it's harder, and they do adapt to the various control devices that are non-lethal, like noise, sound, or visual, scary tactics. And I deal with birds at my house. We have a little vineyard, and just basically have almost given up. But we're using netting that might not be, possible in a very large vineyard type of setting. That is an area that I think it ends up being a case-by-case basis. And in one situation, this was an extreme situation, but up in Alaska, there was an outbreak of Campylobacteriosis that was linked to huge populations of sandhill cranes that were migrating in on the flyway. And there was a pea farm that sold fresh raw peas, and they just were not able to mitigate the bird problem and don't grow in that spot. That's the worst case scenario. But to the extent you can work with wildlife specialists and really look at what your species are and where you actually have intrusions and defecation. They're flying over. That's not the same as if they're in there actually causing damage to plants. And then again, work within the legal, what you can do legally with the birds, and try to reach out to either extension or to the wildlife biologists that are available through government agencies to help mitigate the problem. But I don't have any magic answer. It's really case by case.
Oh, come on, Michelle.
Oh yeah, I mean, we did try falconry, and that can help, but it won't work at every location. So you have to get creative with birds. And
all right, well, thank you very much, Michelle, for your presentation and taking a few minutes to answer the questions here. Appreciate it very much.
Sure, sorry about my technical glitch.
Oh, no problem. It happens. We're going to move on now to our next speaker, who is Dr. Rob Atwell. He's a professor and director of veterinary medicine extension at the School of Veterinary Medicine at UC Davis, and a principal investigator for the Western Center for Food Safety. His research and extension program focuses on the epidemiology of food and waterborne zoonotic diseases and the development of intervention strategies at the landowner scale for mitigating these microbial hazards. Please welcome Dr. Atwell.
Yeah, so thank you guys for all attending today. I appreciate the attention. I think we're all pretty focused on trying to understand this problem that continues to occur, unfortunately, stretching back, several decades. So as Michelle mentioned, this whole question about how does E. coli O157, so I'll focus solely on O157. We could have a whole nother discussion about Salmonella and the non-O157 S-Tecs, but it would take a lot more time than the 20 minutes that we've been given. But what are the key vertebrate sources out there in the environment? And so being a veterinarian, I'm interested in animals, whether they're domestic or wild. And are you a key, and these are important points, are you a key reservoir host? So we find it consistently in you, and from you, this population, we are disseminating out and potentially infecting others. Or are you an amplification host, where perhaps you are repeatedly infected by the reservoir, you amplify it, and as a consequence, you contribute in the environmental cycling. So that could be the same species, or those could be separate species. So I'll talk about some animal studies that focus on this role of domestic and wild animals amplifying or functioning as reservoirs of E. coli O157. Many of you are far more familiar than me with the Salinas Valley, and it's a challenge. Beautifully productive, very innovative farming strategies, very successful. California, we're proud of our agriculture. We do a great job, but we're victims a little bit of our success, right? We produce hundreds of millions of servings each year, ship it throughout the United States internationally. One small problem, and it can be magnified, unfortunately, either in a recall or an outbreak, as we well know. So as Dr. J. Russell mentioned, we love our fresh, crisp salads. We love a good steak, many of us. So we want both to succeed. Challenge, of course, is on the western slopes. We have a little bit of cow-calf operations, pretty extensive, not feedlots in that part of the world, but we have extensive wildlife habitat and vineyards. We have the Salinas River running down the middle of the valley. In essence, a wildlife corridor. You have a lot of woody species. That's a challenge. On the east side, we have very extensive cow-calf operations. They've been there a long time. We also have wildlife habitat that utilize that landscape. A handful of CAFOs. Worried about winter runoff. It's often windy. We have ditches throughout the valley floor that can be effective in moving pathogens down slope from the upper slope locations. Key questions that we keep struggling with. Again, many of us have asked these. Is it infield defecation, this photo of 10 crows, if you can count them real quick? And or is it something in the far distance? Many of us are worried about livestock operations on the hill slopes. And as Dr. J. Russell mentioned, amplification or cattle functioning as a reservoir of O157, and in some unknown mechanism still to be discovered, frustratingly, it moves downslope. And if it moves in winter, it has to over early summer, over spring somewhere intact, and then amplify in the typical late summer, fall outbreak season or recall season. Not always, but an alarming pattern of the late summer, fall outbreak. So if it's that large, because it clearly takes more than a handful of CFU, right, to generate a state or multi-state outbreak, why are we having such a hard time finding it? So these are crazy questions that industry, academics, government still struggle to try and answer. So well over a decade.
If I could interrupt for 1 second, Rob, I'm getting requests for you to move this into full presentation mode so they can see the images larger.
There you go. Sorry about that. Does that do it?
Yes, much better. Thank you.
All right. So about, over a decade ago, we did a longitudinal study. It was a large collaborative project with the industry, both produce and livestock. We worked with the AIS, Dr. J. Russell and myself and others, Cooperative Extension, a whole host of folks worked on this project. I'll just focus mostly on the cattle part. So we followed 8 cow-calf herds, 2.5 years, very intensive study at the time. We would visit the herd at multiple times. If they didn't have the cattle up in the natural setting, we'd have to walk through the range and collect cow pats. So I just want to focus right away on the key results. And this is where I'm going to challenge, and I'm speaking for myself based on the data from these studies, the assertion that cattle are a key reservoir host. So based on this study that tested over 2,000 fecal samples, we generally didn't find very much E. coli O157. Some of these herds outlined in blue, to our surprise, throughout the duration of the study, we never had a single positive. So looking at herd A, that's almost 500 fecals. And you would think that at some point in time, if that herd was consistently infected, we're sort of randomly collecting fecal pats. We're not focused on the healthy or the sick or anything. We would have detected at least one positive. So we were surprised that there wasn't more consistent infection based on the previous feedlot data. So much as what's typically cited in the literature, cited in the press, is based on feedlot or dairy work and not low intensity, few cattle per acre, cow-calf type operation typical of the greater Salinas region. So herd A, C, and H, we were unable to find an E. coli O157. The ones outlined in white we would follow them. Then we would have one herd visit where we got a handful of positives, and then the isolate would disappear. What was really interesting and somewhat disturbing is herd D. We were following herd D, negative, negative, negative. And as I indicate down here in the yellow lettering, all of a sudden 90% of the cattle tested positive. The next time we went back, it had disappeared. So somehow that herd apparently became exposed. The O157 amplified, spread throughout the herd. Cattle were participating in the environmental cycling of this pathogen. And if you were across the way, as Dr. J. Russell shared with the 2006, I wrote it down, 34% of the cattle fecal samples tested during that outbreak investigation tested positive, but that's very typical to the usual negative, zero or low prevalence that we find in these cow-calf type operations. So something happened. There had been a positive pig that a hunter had killed on the property a few months earlier. You could make up a story. Maybe that's what introduced it, or something else happened in that herd. At this time, we still don't understand why. Occasionally, when we do these studies, a herd has 30, 40, 50% of the animal shedding O157. If those are the circumstances, maybe combined with either climate, bad management, or rushing, whatever the circumstance is, that combine and create the conditions for a recall or an outbreak, but its unpredictability when this occurs is what really concerns us. We did take some water samples and soil samples on these ranches at the same time, and it was not that impressive, single digit, low digit % prevalence for E. coli O157. I won't go all into the fingerprinting, but what was a typical observation, and I shared the papers for those of you, you can track it down. I gave them all today, and those can be read. They've been out there for over a decade. But when a herd did become infected, it was very clonal usually. It was very highly related, as if there had been an introduction, it has spread through the herd, and there wasn't a lot of opportunity for genetic change, like it had been around for years and after years. If it had been around year after year, we would have found it, right? We wouldn't have had such a low prevalence as I'm showing here. So the genetic fingerprinting is somewhat consistent with this idea that we get repeat introductions in these otherwise low prevalent or negative testing herds. Contrast that with the wildlife work. Wildlife Services did an awesome job from USDA partnering in this project and testing animals. Feral pigs came in. Other studies have shown feral pigs shedding E. coli O157. So just to be a bit pointed, on average, feral pigs are twice as infected than the beef cattle that we tested in this study. And I'll show you a subsequent study. So it's hit and miss. You never get to test large numbers of wildlife unless they're small, like the blackbirds. You might get a group of feral pigs, a family, but it's hard to get 30, 40, 50 fecal samples, boom, in a day of feral pigs like we can do with cattle. So it's a little bit more sporadic, the data that we generate. Salmonella, we tested a subset you see here in the bottom, and out of about 800 cattle, we found one positive. So we found that really remarkable, that Salmonella was so low in these cow-calf extensive type operations. Wildlife came in at 3 or 4%. I don't want to focus too much on Salmonella because we're talking about E. coli. So what's the problem with comparing wildlife to cattle? You run cattle through the chute or you walk through the field, you can pretty much get as much poop as you want, generally speaking, unless it's a small calf and they've already recently defecated. So if you test a large amount of fecal material, even with a low concentration of E. coli, it's going to test positive every time. So this is a bias when we do these types of studies. So a 10-gram sample, think of a little grape tomato. It's not that much. If we get a smaller sample, now we're talking about a small mammal in a trap, everyone's infected with 1 bacteria per gram, little thimbleful of fecal material. Everyone's infected in my fake study. Suddenly the prevalence is 93%. If it's a smaller animal, now we're down to a ground squirrel or something. Let's say everyone's infected, only 40%. because we're testing so little fecal material, we start missing it. So if you simulate, this is a simulation, why do we potentially see low prevalence in wildlife and we see higher prevalence in large mammals? Humans could be included perhaps. It's because we test such a small amount of fecal material that it fakely makes the prevalence very low. So when you see this type of data, always be aware that if the populations generally shed low concentrations of bacteria, you're going to always underestimate the prevalence in small wildlife and more accurately estimate the prevalence in large mammals that donate a large fecal sample. So we did an extensive tracking program on all of these collaborative produce farms. Thank you very much. That's the only way we get this type of data. We trapped over 8,000 trap nights. About 10% of the time, we nailed a rodent. The vast majority were deer mice. We didn't find that much E. coli O157. The good news, because these were along the dirt roads, along the edges, obviously, the producer didn't want us actually out in the field capturing an animal and prolonging its stay where it may defecate. A little bit higher salmonella, and just if you're ever concerned about crypto, number one concern, these rodents shed very high concentrations of Cryptosporidium parvum. About 50%, if you fingerprint it, is C. parvum, human infectious. So just an FYI, I know cyclospora is more the rage now, but these rodents are loaded. So this is older data, as I said, 10, 15 years old. We redid the study all across California. Thank you very much, as always, to the ranchers who stepped up. We keep your names confidential. To UC Cooperative Extension Livestock Advisors, who helped either sample or coordinate and identifying these cooperative ranches. We want real-world data for these types of studies. In this case, we visited the herd maybe twice. What did we find? Right away, the salmonella, again, was generally absent. It was a different lab this time, same result. So I would not put cow-calf operations near the top of the list if you've had a salmonella hit. At least for me, I would put wildlife much higher, because random sampling indicates, unless something odd has happened and stressed out the herd, that salmonella generally isn't occurring in these beef cattle operations out on range. E. coli O157, we picked up a little bit more. Now we're up to about 5%. Weaned animals were quite low. It may be notable. You would want to repeat this, but non-weaned animals were quite low. The weaned, it jumped up, whether it's stress or what else is going on. They're being handled. They missed their dam. Whatever is occurring, we saw a higher prevalence, about 3-4 times higher in these weaned calves. So let me walk through the results. A little bit more than half of the herds we visited here across California, we just kind of show up when they have the cattle or we have to walk through the field. Again, it's up to the rancher. We didn't detect a positive sample. We only collect about 40 fecals during the herd visit. To the best of our ability, we split it between calves and the cows, 20 and 20. And so a little bit less than a half, we detected a positive. Most of the time, when we did detect a positive herd, the prevalence was in the single digits. But again, just like in that earlier study along central coastal California, 2 herds suddenly had a spike during our second visit. And this is what really disturbs us. Again, we don't know why. And this is where beef cattle operations, if this occurs, bad time, bad management, rainfall event, we connect all the dots. If this random high spike amplification event occurs, it obviously could create the conditions for a recall or an outbreak. So second time we've observed these patterns, the spikiness, the generally low prevalence of O157 and the very low salmonella. So I'm always a little bit skeptical when someone says, well, cattle are the reservoir. And I'm like, well, I don't know about range cattle, about cow-calf in California. I got to follow the data. I'm just not going to agree outright with those kind of generic statements. What about risk factors? These are just associations. These aren't causal yet, but they're compelling. Length of calving season. So ranchers might work pretty hard to keep their calving interval tight for a variety of production reasons. Those were the herds, sadly, that had tight calving intervals that had the higher prevalence. What if I rotate my cattle? Didn't help. Didn't really have any association with the fecal prevalence, at least in these herds. Stocking density. I know those ranchers in the Salinas Valley and those dry foothills will look at, wow, you've got, these are 10 head and I'm counting yearlings, calves, adult animals, all as a single head. It's like, you can put more than 10 animals on an acre. That ain't going to happen in my part of the country. So that's where this super low stocking density of only one animal per 100, 200 acres may be more typical of some operations. So it was just these high density, perhaps irrigated pasture. Again, remember, this is across California and we had some irrigated systems. And then if the cattle have access to surface water, and this is going to be difficult. I'm not sure, and I think Dr. Meyer is going to talk about this a little bit. If we obsess, put bleach in our water troughs with the cattle, if it'll make much difference, but having access to surface water versus groundwater, well water, municipal water, that might get expensive. There was a jump in the prevalence. Then we followed the cattle at our academic herd up here at the Sierra Foothill Research and Extension Center, Browns Valley, followed them for a year. If you have E. coli O157, is it yours? Does it stay the same? This is important because if you participated in an outbreak, unfortunately, not your fault necessarily, just comes and goes. As I showed you, you had a spike event. By the time someone comes out and tests your cattle, and that links to the outbreak strain, you want a high degree of confidence. Well, that's my strain. You're right. That's my strain. Just like, yeah, that's my truck. Yeah, I'm sorry. It was at the crime scene. That's my strain. So again, not going all into the methods, post-fill gel electrophoresis, doing some fingerprints. We tested in March and we got what looks like 5 different strains. You have 5 different fingerprint patterns. Some are more related to others. I don't want to go all into it. You can read the paper. We came back the next month, strain 7 still there. 8 and 9 disappeared, 1 and 2 disappeared. And now we got 10, next month, it isn't stable. Strains come and go. And suddenly 11 and 12 and 13 and 14 were quite different, entered into the herd and we didn't see them again. So traceback investigation, if strain 7 is the one that was associated with the human case, and we were able to get out to your ranch, everyone's cooperating, get to the bottom of it, enhance public health. You're like, okay, maybe this is my strain. 11 and 13, maybe that's my neighbor's strain. So it'd be a bad day if you got blamed for your neighbor's strain, but it hopped across the fence and was just there for a month. And sorry, you were the cooperating individual, but it was actually someone else's strain. So who owns the strain? These are things that make me worry a lot. Why do we care about scat? And Dr. J. Russell also had data. We don't have enough time to share it. If you have manure amended soils and you overhead sprinkle, and do you get fecal splash? Well, what we did is put scat simulating wildlife intrusion in the field. We irrigated sort of a standard 2 to 3 hours. Sure enough, many of the heads of lettuce, about a third of them surrounding that 5-gram stool, were now suddenly contaminated with the strain that we had spiked into the fecal material. Some of the heads of lettuce had quite a bit. And as you can imagine, those heads of lettuce quite close to fresh scat had higher concentrations than if you're farther away. So this kind of gets to that 5-foot rule. If we were all thinking about it, how far does it splash? Well, that's how hard do you rain? How big are your droplets? How much poop do you have? And do you have contaminated furrow water downslope of the scat? Lots of things would dictate the splash pattern. So worst case scenario, these are just some highlights of other studies that I think are important. That last irrigation before harvest, that evening, night, you have wildlife intrusion. They leave a couple gifts behind in the furrow, some scat, some fecal material, and then you harvest, right? And if it's really fresh material, we can get higher concentrations of E. coli O157. How long does it survive? And so many trials done by Dr. Harris and Dr. J. Russell and others who are active in this area of research. We would spike heads of lettuce. What was interesting, some trials were done with water and it dies very quickly. So this is a log reduction, 90% reduction, 90% again, 90, you're going to keep dropping it down to where you only have an infinitesimal amount. So if it's just water, it died very quickly. So these were done in the Salinas Valley. But if you had a little bit of fecal material in the droplet you applied to a head of lettuce, it survived much longer. And more disturbing is this huge variation. Sometimes the bacteria would grow above what we inoculated, and sometimes the bacteria would die. It's these growers at the leaf level that are the big concern, and that can cause potentially human illness if it makes it all the way through the system. So we're not going to believe one study. So we repeat the study in 2012. And why just do rabbits? Let's also do pigs, and let's do chicken poop. And let's do it in 2 different seasons, during midsummer and late summer during the outbreak season. That baby survived really well when the E. coli had access to the nutrients available in that fecal slurry. So we inoculated a high concentration because we wanted to see this decay curve. It started growing, then started dying. Finally, plants are demanding an irrigation event. We irrigate. The next day, again, concerning, it grows. It goes crazy, multi-log growth on these heads of lettuce, and then it begins its death trajectory. So obviously, take-home message would be, yeah, you don't want to harvest the day after irrigation, perhaps, because if you have bacterial contamination on the surface of these commodities, you may experience bacterial growth, and it could be multi-log growth. So giving it a couple days to inactivate and continue its slow decline. If you fit a regression.
We're at time, Rob.
Okay, last slide. Fit a regression curve, you see the same thing. And so obviously, minimizing contamination in the first place allows that die-off to occur that much quicker. Thanks to the crew, and happy to answer any questions.
Thank you very much. And we do have a couple of questions here. Well, we have several questions, so we'll try and work through this as fast as we can, if that's okay with you.
Sure.
First question is, could you characterize or go beyond the prevalence of pathogen and say something about the concentration of the pathogen that was found?
Yeah, a lot of these earlier studies, we did yes/no testing. And in other studies, not the 2 that I cited today, you do concentration. And you'll get a, how should I say it, a bell curve where many of the animals that are shedding, so this would be probably beef cattle in the studies that I'm associated with. Again, difficult with wildlife because you often don't get enough material. But let's say you get a somewhat bell-shaped curve with a tail going to the right. You have a few animals shedding high concentrations who are infected. Most of the infected are down at the 100, 1,000, maybe the 10,000 CFU per gram. A handful, I know we call them super shedders. I don't know if they're consistently the same animals. This is where it gets controversial. If it's a hot strain of bacteria that has the ability to replicate in the host, do you have a stressed host, or is it just the natural course of disease? Many times when you get sick, you shed a little. When you're really sick, you're really pumping out the pathogens and then you recover. But that narrow time when you're at the peak is just randomly when an investigator comes and grabs your fecal sample. So it ends up looking like, oh, there's a couple super shedders. So super shedders can get pretty high, 10 to the 8th, 10 to the 7th CFU per gram. And if you defecate a kilogram like cattle might, or if it's wildlife, to be fair, you have the potential to contaminate whatever's in the immediate environment.
Okay, thank you. Were the random high spikes in the I guess it's cattle set D, in the late summer time frame? And would seasonability be a theory to pursue?
Yeah, so let me hop roll back. We don't have a lot of time. I probably skipped a little bit through the seasonality of that. That one was in September, if you recall, and about half of those hits. This herd, when it tested positive once in the study, was September. D had a September event. F had an October event, right? That high risk time. This herd had a spike twice, not very high because only 6 positive animals. But it happened 2 times, and then we have 1 in April. So about half of these, not a big sample size, occurred during that late summer, early fall event.
Okay, and one last quick question. Did you detect any of this in areas where they were treated with biosolids?
Can't answer that. So we didn't label those environments or farms that receive biosolids and not biosolids. Great question, but that wasn't, unfortunately, part of our study. We did do a project with Monterey. You know, not to touch on sore subjects, to summarize quickly, trying to think what's confidential. You know, widely known there was a sewage spill. We were asked to test that soil quite exhaustively and thoroughly in many, many fields. And it was unremarkable, surprising, given that 1 million gallons of sewage had spilled in an event. But subsequent to that, we were not able to detect really anything unusual in terms of pathogens in those soils. So that's the only thing I know.
All right, thank you very much. Appreciate your presentation, Rob, and taking the time to answer a few questions here. We're going to move on to our next speaker, which is Dr. Gabby Meyer, also of UC Davis. She's the Assistant Specialist for Beef Cattle Herd Health and Production in the School of Veterinary Medicine at UC Davis. Her research projects focus on infectious disease epidemiology, antimicrobial stewardship, and preventative herd health medicine in California beef cattle, with a focus on cow-calf operations. She is also a state coordinator for the Beef Quality Assurance Program in California. So welcome, Dr. Meyer.
Thank you very much for the introduction. So I am, let's see, trying to share here. Good. All right, does that look all good? You can see it?
Yes, looks great. Thank you.
All right, great. All right, so I know we don't have a lot of time, so without further ado, let's just jump in. So I was asked to present on the topic of E. coli O157, of course, reduction in beef cattle, challenges and opportunities. So what I want to present to you today is, I just want to say a few words about E. coli O157 in cow-calf operations. This is not going to be new. This has already been mentioned. But then what are some possible options for reduction, and how do they align with that beef quality assurance program, and how do they fit into normal operations within cow-calf ranches? And just for those who don't know, the Beef Quality Assurance Program, it's a industry-driven certification program that is there to ensure a wholesome and safe product, so beef, of course, and also to ensure animal welfare and health and worker safety. So I think we've heard this now a couple of times, that E. coli O157 in cow-calf operations, it's not really for the range. It's not considered a problem because it's not a pathogen. So when you look at this dam with her 2 calves, you can't tell whether they harbor O157 or not. Their manure doesn't look different. They don't have diarrhea. It doesn't affect their production. So there's really nothing in a typical herd health plan for these herds that would address the shedding of this particular bug specifically. There are E. coli strains that are pathogenic. So for example, calves, when they're newborn, they can get a diarrhea that they can get really sick from. Also, E. coli can cause mastitis. That's typically more the case in dairy cows. So beef cows get that very rarely. So when we study E. coli O157 in cattle herds, it's really for public health issues. So it's considered a problem because it can contaminate the meat. So it can contaminate, when it's on the hide, when feces are on the hide, when animals go to slaughter, it can can contaminate the meat and then cause illness in people who consume that if it's undercooked. Or then, of course, as we have heard now extensively, as an environmental contaminant. So what are some possible options for reduction? So I lumped them together as management, feed supplements, and vaccination. And under the management category, I want to talk a little bit about stress reduction, manure management, and water trough management, if you will. So stress has been shown to correlate or to influence E. coli growth. And from now on, when I say E. coli, I mean O157 or other pathogenic E. coli. So the ones that we're interested in. So there are in vitro studies that have shown that epinephrine and norepinephrine, they stimulate the growth of E. coli. So, epinephrine is the same as adrenaline. So you could say that E. coli 157 is an adrenaline junkie. And there is also animal data that supports that. So down here in this table, you see different groups of calves that have been hauled or transported in a cattle transporter for either a short period of time, 3 hours, or long periods of time, 15 hours. And they were either what's called preconditioned or non-preconditioned. So preconditioned means that the weaning process, or the separation of the calf from the dam when the calf has grown up, that is a really traumatic experience for the calf and is very stressful. And so this preconditioning sort of softens that blow where the calves are still kept on the range after separation from their dam. Also, some of the procedures like vaccination and castration, they're done way before that the transport. So as you can, so here is sort of the list is from the least stressed, so preconditioned short haul, to the most stressed non-preconditioned long haul. And there seems to be some sort of dose response where the ones that are the most stressed do shed more. And so, sure, stress reduction is, probably helpful in reducing STEC, but of course, you have to realize that these calves, these non-preconditioned that were shipped around for 15 hours, that's an 11 on the stressometer for these animals. So that's about as stressed as you can make them. So overall, life on the cow-calf operation, I would say, is pretty low stress anyways. So there is limited efficacy or limited room for improvement. Also, this low stress cattle handling is already part of the BQA program. So every time we give a presentation for BQA, that's part of it. So ranchers are already aware of that tool. Manure management. So we've heard that there can be so-called super shedders that shed large amounts in the feces, and it can survive for some time on pasture, I guess depending on the environment. So in a feedlot situation, you have a lot more options in managing this manure, in scraping the surfaces, in trying to contain that, while on the range, obviously that's not so much a possibility. Also, the manure is considered fertilizer, so it's not even really wanted or desired. What is desired, though, is to avoid manure contamination of feed and feeding equipment, and that is also part of the BQA program, because not only could it harbor E. coli O157, but other pathogens that we don't want to transmit into the feed. And then water trough management. So we know that O157 can survive in drinking water for some time. So one study showed that it can survive for 8 days at lower temperatures. In this case, it was 41 degrees Fahrenheit. So lower temperatures seem to enhance survival of it. But what could possibly be done to reduce the burden in these water troughs? So chlorine is an FDA-approved water treatment, and if you just add a little bit, so 2 to 5 parts per million, so that's 2 to 5 mils of chlorine in a large amount of water, 265 gallons. So that would eliminate, or that would disinfect that water. But the problem with that is that organic material will diminish this effect pretty quickly. So, every time they put their mouth in the water, they have feed in it, they have, a bunch of, other bacteria in it. So it just gets contaminated pretty quickly, leaves that fall into it, dust, you name it. It would be tremendously difficult to maintain this chlorine level that is effective in these water troughs. So even in a feedlot situation, it would at best be difficult, but out on range, it is literally impossible to do that. So that is also, it is straight out of an assessment by FSIS on E. coli reduction measures. So under field conditions, treating livestock drinking water with chlorine has shown negligible effects. So unfortunately, it doesn't seem to really be feasible. Nevertheless, providing clean water is part of the BQA program, mostly for things like nitrates, so obvious toxins that would make the cattle sick. So the next on the list is feed supplements. So there are a lot out there, and they can be lumped into different categories. So we have what's called prebiotics, and these, to simplify it, is basically food for good bacteria. And then we have the probiotics, and that's the good bacteria themselves. And the mechanism for both of those is that if you enhance these so-called good bacteria in the gut, they can crowd out any harmful bacteria that the animals may become infected with. So it's essentially like eating yogurt, where you enhance your microbiome. So the microbiome is sort of a buzzword as of late. That just means that all the microbes that we and other mammals harbor in our digestive system has been recognized as really important for our well-being, for our immune system. And so it's been studied in cattle as well. And so a lot of these supplements are marketed as enhancing immunity, as just promoting good health, but they have also been studied as a way to reduce E. coli O157. And then there's other supplements that have direct antimicrobial activity, such as orange peel or some essential oils. The problem with all of these is that they're designed to be mixed into a mixed ration. So they're designed for the feedlot, where you can design the diet of these cattle, to the dot. While on range or on pasture, of course, they're supposed to eat grass. So, you can try and mix it into other supplements, such as mineral mixes, etc. But the question is, how can you do it in a way that ensures that they're getting enough, and also that these good bacteria are surviving. So that's the problem with the probiotics. They're fresh, they're fed fresh. And I don't know how long they would survive if they were not consumed within a ration, within 24 hours. So I'm just going to give you a couple examples that have been studied. So there one is called Tasco. It's a seaweed. And so in one study, it's shown to reduce O157 shedding in fecal samples by 11% when it was supplemented for 14 days. It was 2% dry matter in feedlot cattle. So if you think a normal cow eats, somewhere around 25 pounds a day. So 2% is half a pound. So, you have to get that into them somehow. I don't know how good it tastes if you don't mix it into the rest of the ration. So there are a lot of questions. So yeah, and the other thing is that because this particular product is designed to be given for 14 days at the end of the feeding time in the feedlot, because that's when they want to reduce it. They don't really care how much they shed up until the very end, because you want to reduce the contamination before the animal goes to slaughter. So they're focused on a very short period of time, while on range, you would want to reduce it, all year long or during the summer, or at least for extended periods of time for sure, which can become costly. This is another example of a probiotic. So this one is Lactobacillus acidophilus and Propionibacterium freudenreichii. So this is one that so bovamine defend is an example for that combination. And this has been shown in a few studies to be beneficial. So this is one result, or this is results from one study. The black column is the control group, and then these other columns are various levels of supplementation. And you can see that 2 out of 3 did reduce the prevalence of shedding significantly. However, there was one group where they didn't see a difference, a statistical difference between the control group. So in the discussion, the authors mentioned that, well, there was an animal that shed a lot, so a super shedder, so probably that, led to this result. But that just underlines that, well, these things can work in certain situations, but then if there is a super shedder, then they don't, all of a sudden they don't work. So, and then there is, this is a very new study from 2021. This is a different product, BacterShield. This contains Lactobacillus acidophilus and KCI. And they fed this for 60 days before sampling in 2 commercial feedlots in Kansas and Nebraska. And here they didn't see any differences. So they didn't see differences in prevalence of shedding in either feedlot. And then in terms of prevalence of super shedding, actually the treatment group in one of the feedlots had higher prevalence than the control group. So that underlines it's important when if you wish to use one of those products for this purpose, that not all of them seem to work the same. And so it's important to do the homework and find out whether it's possible to deliver it, whether it actually works, and then of course, what is the cost. And then finally, the vaccine. So there is one commercially available vaccine in the US. It's this Suretia Coli bacterial extract vaccine with SRP. It is now fully licensed, even though this image of the bottle says conditional license. It's now fully licensed. It means the USDA is convinced that the product is safe, pure, and efficacious. It also has been designed for use in feedlot. However, it is not widely used, although it has shown to be efficacious. For the same reasons that we've already talked about, it doesn't make cattle sick, so giving it doesn't give the feedlot any advantage. There's no difference in the production. It is only a public health measure, and it just hasn't, for the feedlots, worked out that this is where they're going. So, but how does this vaccine work? So there are surface proteins in E. coli O157 called siderophore receptors and porins, and those help the bacteria to sequester iron from its host, and that's necessary for its survival. So if you give the vaccine, then the host develops an antibody response to these receptors. And when the bacteria invade, then those antibodies bind to those receptors and starve the bacteria of iron, and then it will die. So the vaccine is labeled to be used in cattle 5 months or older, 3 doses, a booster at 2 to 4 weeks and 1 6 weeks after the initial dose. It has a 60-day slaughter withdrawal time, and it costs $2 per dose. So how well has this been working? And again, of course, we only have data from feedlots. So there's one study where they did 2 trials with a large amount of cattle, so over 1,200 cattle. The first trial they did, they only gave 2 doses, 21 days apart. And what you can see is that in the end, they didn't see a difference. And again, in feedlots, that's where what they're looking for. That's where they want to see a difference. So they went to 3 doses. However, with the 2 doses, they still saw a 50% reduction in the prevalence over all days. So when you combine all these sampling days, they found that in the vaccinates, so the dark circles are the vaccinates, they had 50% fewer animals were shedding than in the controls. With the 3-dose regimen, they did see a difference in the end, and that's what they were going for. And they found that there was an 85% reduction in prevalence, so number of cattle that were shedding. And then they also found a big reduction in concentration in feces, so 98.2%. So those that were shedding a lot fewer than those in the control group. So overall, I would say that these numbers are encouraging. Now, how does this work into a cow-calf operation schedule? So we don't really have time to go over the cow-calf production cycle. So this is but just briefly, this is basically, this represents a year in the life of a cow-calf operation. So everything is on a schedule. There's times for calving, for breeding, and then times when the cows are supposed to be pregnant. But the thing is that these cattle are only handled occasionally. And as Dr. Atwell has shown with his data, that if you have higher concentration or you handle cattle more intensely, that also led to more shedding. And so it's just something where you introduce stress, you have the potential for accidents, for injuries. So cattle are only handled when it's necessary. So there's only a few times of the year when we actually want to, or when cattle are typically handled during the year in a cow-calf operation. So I just want to mention that we do have a project studying this vaccine in the cow-calf operation at this moment, where we're giving 2 doses of the vaccine to pregnant cattle. So at times when they would, or when they do normally handle them, where they give other vaccines. And caveat, the product has not been tested in pregnant animals, so we're doing it. One of the reasons is that we want to see whether it's safe. We want to compare the treatment and control groups for prevalence of shedding, as well as the quantitative assessment of the shedding. And it's important to understand that this is a very small pilot study. So you saw that in this other vaccine study, they had over 1,200 cattle. We have 60. So it's unlikely that we will see significant differences between the groups. But it's mostly to see if there are any trends, what we could do better next time. And we hope to move these trials to commercial herds in the future and see how well this vaccine does. I just wanted to leave you also with this quote. This is from the author of the study of the large vaccine study, and they said that it is unlikely that absolute elimination from the live animal will be possible. So even though we do have some tools available, I hope I was able to outline some of the caveats that come with using these tools. And so with this, I will take your questions.
Great, thank you, Dr. Meyer. And the first question is, will cattle still be considered natural if these feed supplements are utilized?
Good question. I'm not 100% sure. I think that's probably a question for the technical service of the manufacturers of these products. None of these are antimicrobials. They're not, well, I said they have antimicrobial activity, but they're natural products. So the orange peel, the oils. I mean, none of these are products that, I think they're all pretty benign, but I would double check with the manufacturer for sure.
All right, thank you. And then real quick, what's your estimation of feasibility of implementing this in a cattle operation, or more of a widespread application for cattle operations across the state? Is this something that's actually feasible if it proves to be as successful as everyone is thinking it will be?
Are we talking about supplements, or are we talking about the vaccine?
The vaccine, I'm sorry.
Oh, the vaccine. Well, I mean, we're doing it because of course we're hoping that it will be successful, but the truth comes out, the truth, science will tell, and the truth comes out in the end. I mean, there are a lot of unknowns that we will, that we don't know yet. So the feed is different. Right now we have a pretty wide time frame between the initial dose and the booster shot. Is that going to be too wide? Is it not going to work? So do we have to go to a 3-week booster? Or do we even have to give 3 doses? So all those things we don't know yet. I mean, if we have to give 3 doses 21 days apart, I would assume that that there's going to be very little acceptance, especially in pregnant cows. So I don't know. I mean, I'm hoping that we can offer a tool, but it remains to be determined how effective it is going to be.
Okay, great. Well, thank you very much for your presentation. Appreciate all the great information here. And with that, I'm going to wrap up this webinar and want to thank all 3 speakers for their information and their presentations here. We heard about wildlife movement and E. coli harboring in wildlife and how that transmits. Thank you, Dr. J. Russell. We appreciate the 2 I can't even say it. Sorry. The 2 surveys on E. coli that were presented by Dr. Ratwell. Thank you very much. And also thank you, Dr. Meyer, for a review of the challenges and the opportunities for addressing E. coli in beef cattle. So with that, we're going to wrap up our first webinar here. Remember to register for next week's webinar and then the following week. So that means July 7th and then July 14th. Those webinars will start at 9 a.m. each of those days. So please make sure that you register and get the links for both of those. And I also wanted to remind everyone that we did record this webinar, and we will make that link available for a YouTube channel view at a later date. So look for that as it comes out. We can't give you the link now because we have to close the webinar first to get the link. So it's one thing after another here. Anyway, I wanted to take time to thank you all. We had a great number of participants in this webinar. It's great to see a lot of interest in these topics, and we hope that continues with the next 2 webinars. And again, thank you for joining us today, and that concludes this presentation.